old question paper. I think we should try to do it. And it's a theoretical problem. And I hope you remember we have solved a few questions on imprinting status before. And I would like you to do that as well. So take a screenshot of it, and you can circulate it to your friends as well. And try to, maybe I will take a screenshot of it and then see. No, it won't cover. Yeah, I will try to take a different picture again, and then I will do that. So I want you to discuss it among yourselves and come up with answers. Okay? Yes. Rishi, did you upload any questions on that? I gave
No,
an
sir.
assignment.
I was sick the week, and
Okay, that's all right.
but I had one. I was in a biology society group, and they had a discussion like.
What discussion is it?
Yes, just a minute, sir.
No, what meeting is it?
It wasn't a meeting, sir. It's a WhatsApp group. It has researchers and scholars from around the world, and
Okay.
it's a biology society. The name itself is
Okay,
TBS.
biologists. Okay.
Uh, so there was this thing. So a person asked if we would be able to distinguish paternal and maternal chromosomes using epigenetics.
Okay, that's
So
nice. Mm-hmm.
there was this discussion, and it felt quite relatable to the subject. Maybe that would be a
Yes.
20 mark, probably.
What would be the answer?
We could use, yeah, we could use epigenetics because we can find which are repressed and which are expressed.
No, but it is with genomic imprinting alone. If the gene is not genomic imprinted gene, then we might not be able to find it. Is that right?
But usually we have imprinting, right? Because most
Yes,
of our
yes.
genes are not functional, and they must have been imprinted because they are
Uh,
not
no,
functional.
we cannot say like that. Um, they must have, well, paternal, and there are common genes between paternal genome and maternal genome. Many of them are common except for the sex chromosomes. And some genes are functional. Some alleles of the maternal alleles are functional, and some of them paternal alleles are functional. And it could be because of methylation, DNA methylation, or epigenetic modifications, and so on. You cannot say that gene is it is not functional at all, but it's not expressed. Yes, what you said does make sense. But the number of genes that are controlled by genomic imprinting are very less. Okay, relative to the genes that are not under genomic imprinting. So what you said is correct. If they through epigenetics, and if the question didn't specify, we can identify based on the imprinted genes. Yes.
But shouldn't we able to, you know, take out the chromosomes just based on imprinting because they are on the same strand? So will it not be
No. How will
easier?
we take out the chromosome?
This is just classification, right, sir? Paternal and maternal. So if we find a paternal imprinting on a certain chromosome, we'll classify it as maternal,
Yeah. Okay.
paternal, and
Classification. Yes. Yes. We can do that. We can do several molecular techniques are also available to find out
they suggested
methylated histones.
VNTR and
Yes.
microsatellite markers.
That
They
is not
suggested
epigenetic, right?
uh, no, they
They're
suggested that would
genetic
be much
methods.
easier than epigenetics.
Yes, yes. Those are not epigenetic ones. They're just genetic ones. Genetic markers, you can call them as. But the
Oh,
question says,
but
uh,
methylation itself can be a marker, right?
which one?
Methylation itself can be a marker, right, sir? So it can serve as both imprinting and also a marker.
No. What its purpose is, yes, genomic imprinting wise, if we already, it is established that these genes are, this allele is expressed, a particular allele is expressed from paternal allele and not the maternal allele. And if you have means to identify that, DNA methylation status, then you can say if it is maternal or paternal. That is possible. Okay,
Yes,
but
sir.
usually more definitive one would be the genetic markers. But the question, somebody asked a question whether you can do it or not. So then what your
Actually,
answer
it
is?
started out from another basic question. So, there was a child, and there is a case, and they have to find if the child belongs to the that couple, that father and mother, and
Okay.
that's where it came from. And someone suggested you could use epigenetics, and
No, there I don't think it is a good method. Neither can epigenomic, say genomic imprinting cannot be used for that purpose, because we take that allele from any female, and if it is if it is genomic imprinted or methylated maternally, it would have been methylated. So their epigenome may not have such a drastic or conclusive. It may not be such conclusive one. Then the other person,
it
whoever
might
has said,
not be specific for that woman.
yes,
Instance.
yes, it's difficult to say because
So we
epigenome
can
can be
find if a
changed.
person is a clone or not a clone based on.
No, that is also difficult. Epigenome can be changed, in the sense it has environment can have an influence on the epigenome. So we cannot use it for diagnosis as much. And there we have to go
So,
with the
but
genetic factors.
say a person lacks maternal or paternal imprinting, we could say that he came from a single parent. So uh, that
No,
would possibly
we
mean.
don't have the technology for uniparental organisms yet.
Oh, okay.
Yeah.
It's just an assumption.
Yeah, that's fine. Every thinking is still at thought. So we know about dosage compensation, other things we have learned in those genes. If we learned about androgenetic zygoids and gynogenetic ones, they don't give rise to viable organisms, or they have, even as cells itself, they have some deficiencies. It's because twice the dosage of some genes and lack of some expression of some genes. Okay. Whereas genomic imprinting in a biparental organism would balance that out and make what we think is we assume as normal. Yes, I don't think epigenome using epigenome in that case is a good idea for diagnosis purpose, a diagnostic or settling parental disputes and other things.
Yes,
Yes,
next.
okay, that's good. Good discussion. If you should have weighed in also. One of the problems is, say, biotechnology students study genetic engineering, and we don't have it in your syllabus at this time. I mean, we have it as an elective, recombinant DNA technology in medicine, which I take. It comes in 7th semester. So there we try to learn, not, but yeah, it's up to you. But you can also do many courses are there on Coursera, which you can do and gain many much more knowledge, especially if you are going to stick with biology side of, and you're trying to planning to go for research in the future. It's very, very important to study genetic engineering right after molecular genetics, kind of, because it is the application of genetic information, and then we are trying to better the cells, better the living organisms, or get something good out of using living organisms. In that case, we have to manipulate the genetic information, and the techniques, all those techniques are they come as genetic engineering or recombinant DNA technology. So maybe you have to wait until 7th semester to study that.
Do
Okay.
we have molecular genetics lab, sir?
I am unsure. Maybe when you come back, we may have those labs. Yes, in that lab also we might. We learn about plasmid DNA isolation, genomic DNA isolation, protein electrophoresis, and other things. I hope when you come back, I think Shastra will try to make everybody do all the labs. That's always been the policy. But when and how, it's little tentative. We don't know what the government
Yes, I just
says.
wanted to know if we'll be able to do those stuff. Like,
I think you will be able.
yes.
You may have to do actually. I think so. I'm unsure. It's also difficult to accommodate everybody in that one month, right? So it will
Yes.
depend on the possibilities. Yes. Yeah. Mm. I usually conduct that lab as a molecular biology lab. Yeah. Okay. Can you please solve this problem and try to upload? You have anything to say, Ritika?
Sure, no, sir.
Okay. Mm, I hope there are no mistakes in the question, because just have a look at it and try to solve and upload it. Okay. I will put it on Google Classroom now. I'll put two questions. One is this one, and there is another question that is, do you remember transgenic animals? Did we discuss transgenic animals?
Yes, sir, we did.
And in that we have made a DNA construct, and we discussed about what are the significances. I don't have my graphics tablet here, so I won't be able to write as much. But I hope the objective is to put knock-in, that means insert gene Z between gene X and Y. So the genome has something like this. It has gene X and it has gene Y. The objective is to put in gene Z, okay, and nowhere else. So how would you? One of the researchers has designed a DNA construct like this, and we need to figure out if it is correct and what is the significance of why these things are put here. Gene, we put gene Z here so that because we want to integrate it here. Why do we have? What are these arrowheads here? Anybody?
Uh, used for site-specific
That's
recombination
right.
sites. Yes.
That's right. That's correct. Absolutely. So you need to think about, yeah, you need to reason why do we have, where do we have, and which orientation it is, and how it should be, whether it is right or wrong. And NPT, do you know NPT?
Used for selection.
That's right. Neomycin phosphotransferase, used for positive selection. And we have TK, that is thymidine kinase gene. So that also we should try to know. I mean, here you are evaluating somebody's approach. Okay, that will require fantastic skills to do that, and if you are able to do that, absolutely, it's very good. You're all allowed to discuss among yourselves and try to solve these two problems. I will post these in the Google Classroom now. Okay, do that, and please encourage others also to do. You are only 2 people out of 11 or so. Try to ping them and try to discuss uh, the questions and see if you can answer. Okay.
Sure, sir.
Yes.
Yes, sir.
Yes. Okay, then I will terminate the recording now at the classroom, and please try to solve them. Okay. Thank you.
Okay. Thank you, sir.
Thanks.
Thank you, sir.